Jo, vodu tady skleničku máš. Super. Jo. A můžeme pouštět tam tudy. Nebo to je jedno. Jo. Tak můžem? So good morning, everyone. It's past 9 o'clock. Looks like that many people got tired after the beer yesterday. But thanks to those who came for the plenary talk. And it's my great pleasure to have Antonin Machac here today. He is kind of my friend from a long time ago. We met for the first time in Papua New Guinea in 2008 at the course of the field Tropical Ecology, which our university organizes and sponsors every second year 10 students to go for a month to Papua New Guinea. So that's where I met Antonin for the first time, and he was that kind of guy who is going around the forest with a hypothesis and really focused on his research while we were just enjoying ourselves with the fruits and drinks. And after Antonin successfully finished all his studies, he went for the world tour for his postdocs, and he stayed in Denmark or worked in Denmark, in Canada, in America. And just recently, he returned back to Czech Republic. He started to work at the Academy of Sciences, but not here, in Prague. And he is establishing his research group here. So welcome, and the floor is yours.
Thank you very much, Katka. Thank you very much for the invitation. It's a pleasure to be here. I realized that this is a morning talk and the poster session was running a little late, so I really appreciate you joining in. The talk will be on why are the tropics mega diverse? This is one of the fundamental questions in biology. And this question, why are the tropics mega diverse, goes all the way back to Humboldt. When Humboldt was about my age, he was traveling the New World, especially the neotropics, and he noticed on his journeys that there are many more species in the Amazon of the tropics compared to the temperate of the Europe. So there's many more species in the tropics compared to the temperate. He also noticed that there are many more species in the lowlands compared to the highlands. And these conspicuous patterns of diversity were later confirmed across different continents, the New World, Africa, Southeast Asia, but also across different mountain systems, such as the Andes, the mountains of Central Africa, or the Himalayas. These patterns of diversity were reported time and again across different organismal groups. Today, we know these patterns as the latitudinal diversity gradient and the elevational diversity gradient. And because these conspicuous patterns are so common, they inspired a number of hypotheses. Over the decades, 3 hypotheses have become particularly prominent: the energy limits, the diversification rates, and the time for speciation hypothesis. The energy limits postulates that the tropics are mega diverse. The lowlands and the tropics are mega diverse because there is a lot of energy for the coexistence of many species. The diversification rates hypothesis states that the tropics are diverse because they accumulate species fast. The intrinsic properties of the tropics facilitate speciation, suppress extinction, species accumulate fast, and that's why there are so many species in the lowlands and in the tropics as compared to highlands and the temperate. And finally, the time for speciation hypothesis states that the tropics are diverse because they are old. There was a lot of time for the accumulation of species. Now, there are some challenges associated with these explanations and with explaining tropical megadiversity in general. For example, each of these three explanations has been supported by empirical results and by theory. So it is not clear that one of these explanations would be better supported than the other one. It's also the case that the explanations are hard to disentangle because their effects overlap in the tropics. The tropics tend to be old, there is fast diversification, a lot of energy for species coexistence, so it's difficult to tease apart the effects of time, energy, and diversification. The next challenge is that there are only two latitudinal diversity gradients, the southern and the northern hemisphere. So when we study the changes in species diversity across latitudes, we don't have as many replicates as we would like to have. And finally, the relative importance of these three explanations and the possible interactions between the explanations remain relatively unexplored. As a result of these challenges, a synthesis has been elusive. We are lacking a synthesis which would integrate these different empirical results and these different explanations to give us a unified answer to why are the tropics so diverse. Given that we lack a synthesis, it has been difficult to advance the theory of biodiversity, to reconcile the conflicts in empirical results, and the lack of synthesis also limits prediction. We cannot anticipate changes in diversity or efficiently conserve diversity. In my talk today, I will argue that maybe there are some promising research avenues which could lead toward the synthesis. And we will explore these 5 points, 5 potential ways to move towards the synthesis. We'll see that maybe we can study the explanations together, that we can investigate the interplay of mechanisms behind the explanations, focus on diversity patterns in general, work across scales, and link the diversity dynamics to the microevolutionary dynamics that generate new species. Now, maybe by taking these lines of research, we might be able to integrate these 3 explanations. I will start with a study that some of you are familiar with because I presented it to those of you who are from České Budějovice before, because it nicely connects with the other studies as well. So in this paper, I attempted to advance such a synthesis. Can we maybe integrate these 3 explanations together? As the model system, I used the very system that inspired Humboldt in the first place, the New World, and especially the neotropics. The neotropics are a well-defined evolutionary arena. We see that in the neotropics, there is the highest known concentration of amphibians, reptiles, amphibians, birds, and mammals, these 3, but also many other hierarchies have the highest concentration of species in the neotropics. There is also the vast areas of the Amazonian rainforest, which provide a lot of energy for the species coexistence, so many species can coexist. The biomes within the neotropics have had a long time to accumulate species. And finally, the Andes, which are over here, might have acted as a diversification pump, right? So they generated species. In other words, in this evolutionary arena, in this region, all of these explanations seem to operate. They come together to generate a biodiversity hotspot. And because they all operate within this system, this might be a good system also to try to disentangle their respective effects. As the model system, I used 5 groups of birds, which you see depicted over here. The ovenbirds, antbirds, tanagers, tyrant flycatchers, and hummingbirds. These are altogether 1,500 species of birds, which differ markedly in their biology. We have the hummingbirds, which are specialists living high up in the mountains with fast metabolism, they're nectarivores. On the other hand, the tyrant flycatchers, for example, are relatively generalist species. The antbirds and the ovenbirds have very idiosyncratic nesting and foraging strategies. So we have 5 clades of birds with very similar diversity patterns, but very different biologies. To evaluate what drives the diversity of these birds in the tropics, I used the geographic information systems, and I analyzed community phylogenetic structure. The community phylogenetic structure captures the relatedness of species within the community. And maybe from this relatedness, under some assumptions, we can estimate possible competition between the species. I also estimated diversification using phylogenies. So here the red clades diversify, they accumulate species very fast. And of course, regressions and some null models. Time was captured using the age of the fauna within the region, right? So time serving as a predictor of diversity within the region was captured as the age of the fauna within the region. And I also used some ancestral reconstructions using biogeobears. Diversification rates were estimated using BAM and DR, and climate and energy limits using climate and environmental productivity. So these are the predictors which we use to explain regional diversity. Let's have a look at the results. The results are over here. We have time, diversification, energy, and the amount of variation in regional diversity that they explain. The results are depicted for 5 groups of birds. And you see that all of these explanations are supported. All of them contribute to some extent to regional diversity, but there are differences in the magnitude of effects, right? So the explanations are differentially important. Where energy limits seem to play the dominant role, they explain over 40% of the variation in diversity. However, diversification also seems to play a role, and even time, which explains the least, still explains about 15% of the variation. It's also interesting that not only do we see these differences in the magnitude of effects, but these differences are consistent across the 5 groups of birds. So this could be a possible conclusion here, right? We evaluated the 3 explanations side by side. We found that each of them is supported. They are differentially important, and these differences are consistent across clades of birds with very different biologies. But maybe we can take this a little bit further. Maybe we can study the interactions between these explanations as well, right? Like what are the mechanisms through which maybe diversification changes over time as a function of energy. We can use hummingbirds as the test case to study the interactions and then look at the other groups as well. In hummingbirds, we see that most species are concentrated in the tropics, especially in the Andes. And when you look at hummingbird diversification on the phylogeny, you see that these 2 clades stand out in terms of their diversification, the bees and the emeralds. Now, you might expect that these 2 clades, the bees and the emeralds, are the Andean clades because they accumulate species very fast. But that is actually not the case, right? When you project the results, the diversification rates onto the map, you will see that these clades are North American. So there is fast accumulation of species in the species poor and newly colonized North America. Whereas in the species rich and long colonized South America, the accumulation of species is relatively slow. So we see that there is a negative relationship between species diversity and the rate at which diversity accumulates. Here you see the same relationship depicted as a scatter plot, where diversification within the region declines as regional richness increases. It's interesting that the diversification within the region also seems to correspond with community phylogenetic structure. Namely, we see that there is phylogenetic overdispersion in the tropics and phylogenetic clustering in the temperate. Now, these are patterns in the data, but maybe we can speculate a little bit that this could be an indication of competition between the many species that coexist in the tropics, whereas in the temperate, we see radiations. The species are closely related to each other. This relationship also holds when we account for some null modeling. Now, these are the results for hummingbirds. But when we look at the results for all 5 groups, we'll see that we find the same relationships, right? In all 5 groups, diversification declines with regional richness, and it corresponds with the patterns of community structure. So for example, in tyrant flycatchers, we see that most species are in the tropics, but new species accumulate in the temperate. These were the results across the different latitudes, the temperates and the tropics. Now let's have a look at the results across different elevations, lowlands to highlands. Again, we can start with hummingbirds, and we see that the number of species of hummingbirds declines with elevation. Most of the hummingbirds are in the lowlands, few are high up in the mountains. Interestingly, regional diversification increases with elevation. So new species accumulate high up in the mountains, not in the lowlands. And this pattern becomes even more pronounced when we divide the data into North America and South America. So you see that the fastest accumulation of species occurs in the highlands of North America, right? So it's the species-poor highlands of the species-poor North America that accumulate new species the fastest. And again, there is some correspondence with community phylogenetic structure, potentially indicative of competition in the lowlands and radiations in the highlands. These are the results for hummingbirds, but we again find the same results for all 5 clades. In all 5 clades, the number of species declines with elevation, the rates of diversification increase with elevation, and this corresponds with community phylogenetic structure. So we saw that we have the 3 explanations, and maybe there is an opportunity to integrate these explanations based on the results that we have empirically seen in the New World and the Neotropics. Namely, we saw the following patterns in the data. We saw that each of the 3 explanations is supported in the Neotropics, but their effects vary consistently with magnitude, with energy limits having the strongest effect. So all 3 explanations are supported, but they are differentially important. We saw that species richness is high in the long colonized regions, but contrary to the traditional view, these species-rich regions show surprisingly slow diversification. Right? Regional diversification also seems to correspond with species relatedness in the region beyond the expectations of the null model. So these are the patterns, these are the correlations that we see in the data. Now, maybe we can speculate about a possible process that could have generated all these patterns simultaneously. One such process, which I am proposing here, might be that the species accumulate at decreasing rates as regional richness increases, whereby the community structure indicates possible signatures of increasing competition. Despite these slowdowns, so the accumulation of species slows down, but despite these slowdowns, regional energy does not seem to set a hard ceiling on regional richness. And we find that even the most species-rich regions, such as the lowlands of the Amazon, right, even the most species-rich regions continue to accumulate species. However, the newly colonized and the species-poor regions accumulate new species faster. We saw that this interplay of mechanisms is not captured under any of the 3 principal explanations. And yet, it is remarkably consistent, and it emerges repeatedly across multiple taxa, covering over 1,500 species of birds. This interplay also holds for the latitudinal diversity gradients at the continental scales, but also across elevational diversity gradients at the regional scales. So why are the tropics megadiverse? It seems that at least in the case of neotropical birds, the diversity patterns result from an interplay of time, diversification rates, and energy limits. Going back to some of the ideas of how we can potentially advance a synthesis. Well, we saw that in this study, we can maybe practically implement the studying the explanations together. We also investigated the interplay of mechanisms invoked by the respective explanations. And finally, there was focusing on diversity patterns in general. So diversity patterns here were studied across latitudes, across elevations, across different taxonomic groups. Let's stay with this last point, focusing on diversity patterns in general a little bit longer. This is a topic that we aimed to explore in a paper with my Brazilian colleagues. Specifically, we focused on the exceptions to the rule. We focused on clades that show inverse latitudinal diversity gradients. right? So there are some clades where most of the species are outside the tropics and few species in the tropics. So they show an inverse gradient where the diversity increases from the tropics toward the temperate. Now, it's important that the three explanations, time, diversification, energy limits, these explanations are defined in a universal fashion. This means that in principle, at least, they could explain any diversity gradient. So for example, when the clade has most of the species in the temperate, the diversification hypothesis postulates that the species-rich regions accumulate species fast. So within this clade, diversification should be fast in the temperate. Right? So in principle, we can explain both the classic and the inverse gradients using these explanations. However, these explanations have rarely been studied outside of the domain within which they were defined. Right? So we typically study them using the classic gradients, and it's not clear if they can actually explain the exceptions to the rule. So let's put them to the test and see if they work. This has at least twofold benefit. The first one is that the effects of the explanations tends to be conflated in the tropics, right? It's difficult to disentangle the effects of time, energy, diversification, because the tropics are long colonized, many species, high energy, and so forth. But maybe when we focus on the exceptions, suddenly in the temperate zone, the time, diversification, and energy become decoupled. So we can better tease apart their individual effects. The second benefit here might be that we can learn about what causes diversity gradients in general, rather than the subset of the famous gradients. You get a more general knowledge of what causes the gradients. As the model system for this work, we used vertebrates whose diversity peaks outside the tropics. And specifically, we chose salamanders, albatrosses, geese, seals, cetaceans, and rabbits. So altogether, these are about 800 species. And you see that these clades differ significantly from each other. Like there are ectotherms, there are endotherms, there are marine species, and there are also terrestrial clades. We use similar methods as in the previous case of neotropical birds. So we studied diversification using BAM and DR. Time was captured using phylometrics and biogeobears, and energy limits using climate and environmental productivity. It's a similar design. Just to give you an idea of what these gradients look like. Here in geese, for example, we see that most species are in North America and Northern Eurasia. There's a similar pattern in rabbits, right? Most species in North America and in Eurasia, or in salamanders, where most species are also in the temperate, but you see that the diversity is concentrated in North America. And even though the temperate zones over here in Eurasia have been colonized, they have much less species than North America. So it's the same climate, it's the same environment, but the differences in diversity are significant here across the different continents. What did we find? Well, by looking at the results here, you will see that the findings vary significantly across the clades. In albatrosses and geese, for example, regional diversity seems to be explained by diversification. In cetaceans, the regional diversity seems to be explained by regional energy. But in seals, all three explanations seem to be comparably important. So we find very different results, very different explanations across the 6 clades. There are more details shown here in this variation partitioning, but altogether the results show that different mechanisms can generate very similar diversity gradients. Right? Different mechanisms can generate similar diversity gradients. These are the same results depicted in a different way. So in black, we have, the black lines are diversity, time is the blue, diversification is red, and energy is yellow. And you see, for example, that in albatrosses, there is a close correspondence between regional diversity and regional diversification, the black and red lines. Whereas in salamanders, there is a correspondence between regional diversity and time. We can have a look at the results for salamanders in more detail, for example, and we see that most species are in North America. There are some species in Eurasia as well. And when we look at the diversification around the world, we see that the temperate regions, which have most of the species, again, show slow diversification. Whereas the tropics, which have relatively few species of salamanders, accumulate species very fast. So it's a similar negative dependence of diversity and diversification. This might be further elucidated, the story of salamanders might be further elucidated when we do the biogeographical reconstructions, right? So we use the biogeobears to have a look at where the salamanders originated. And you see that it's very likely that the salamanders originated in the temperate, probably in North America, and then they spread toward lower latitudes into the tropics. So they were spreading toward the tropics, slowly accumulating species. So there are many species in North America, but once they hit the tropics, the accumulation of species accelerates, right? The species start to accumulate faster, as we see from this diversification map. However, there hasn't been enough time to accumulate many species in the tropics yet, right? So even though the species accumulate fast in the tropics, there are relatively few because the tropics have only recently been colonized. I would therefore summarize this study saying that similar gradients might have different explanations. And we saw that even here, none of the explanations in and of itself can completely account for any one of the gradients. To be more specific, we found that diversification explains high diversity in geese, albatrosses, cetaceans, or rabbits, but not when the region has been colonized only recently, which was the case of salamanders. We saw that regional energy correlates with diversity, but diversity still shows cross-continental differences due to historical legacy. So there was also the case where we saw there are more species of salamanders in the temperate zones of North America than in the temperate zones of Eurasia, presumably because the salamanders historically originated in North America. And our results also suggest that species continue to accumulate everywhere. We find diversification at present significantly greater than zero all over the world. So species continue to accumulate everywhere. One of the conclusions or propositions for further work in this paper that we mention is that maybe instead of searching for the holy grail, whereby one explanation accounts for any latitudinal diversity gradient, it might be more useful or potentially fruitful to investigate the variety of mechanisms whose interactions unfold over time to generate both the classic and the regular gradients. Right? So instead of searching for one explanation that explains latitudinal gradients, maybe we can study the mechanisms that unfold over time and explain both the regular and the inverse gradients. One of the potential concerns here, in the case of this study, might be that these are results for taxa which are by definition the exceptions. They're the exceptions to the rule. They're somehow idiosyncratic. And these clades that we have seen have hundreds of species, which is not few species, but it's still relatively small clades. So it might be that what happens in these clades does not really reflect what happens in nature generally. To address this concern, we conducted a study where we investigated biodiversity in the case of fungi. So it's a very different taxonomic group, and it's a significant taxonomic scope as well within the fungi. Some of my colleagues have samples of fungal biodiversity from all the world's continents, which span all the world's biomes, right? You see the samples depicted here. So what they did was that they've taken soil samples in a standardized way at these different locations, and then you send the soil samples into the lab and you sequence them. Once you have the sequences, you evaluate the divergence in the sequences so as to define not necessarily species, but some comparable operational taxonomic units. So the sequencing gives us some idea of the relative differences in biodiversity across the different samples. These samples included very disparate groups, including Basidiobicota, Ascomycota, Cryptomycota, very disparate taxonomically, but also ecologically. There were saprotrophs, pathogens, ectomycorrhizal fungi. And when I modeled the biodiversity for the microscopic fungi, this is what we found. Right? So we see that there is relatively limited biodiversity of fungi in the tropics. In fact, species seem to be concentrated at high latitudes in North America, Eurasia, at high latitudes in South America. So we see that there is an inverse latitudinal diversity gradient, presumably in fungi, which is a taxonomically relevant and large group. It therefore seems that there might be entire domains of life that might define the classic latitudinal gradient. So it's not necessarily something that is idiosyncratic, but it seems that even like large taxonomic groups might show a gradient which does not have most of the species concentrated in the tropics. This further underscores the need to search for general principles that govern diversity gradients, both the classic and the idiosyncratic or the reverse ones. That was focusing on diversity patterns in general. The next line of research, which might potentially be fruitful and bring us closer toward the synthesis, could be working systematically across scales. Two concepts of scale are very widely recognized in ecology and evolution, and that is the temporal scale and the spatial scale. And we routinely study biodiversity over different geological periods, in Cretaceous, in Cenozoic. We also study biodiversity across different spatial scales, locally, regionally, at the continental scales. But what is relatively overlooked is the phylogenetic scale. The notion of phylogenetic scale has been around for a long time, but it has never been formalized. And this is what we attempted to do in a paper that followed from my dissertation, where we try to formalize phylogenetic scale and define it in a manner which is rigorous enough so that we can use phylogenetic scale in empirical research. Just to give you an idea, I'll briefly go over the definition. And scale is always defined as a hierarchy, right? So scale is a hierarchy where the units of analysis are nested within each other. So when you think about scale this way, it becomes intuitive that phylogeny is very suitable to define scale. In the case of spatial scale, we distinguish between grain and extent. So grain is the smallest unit of analysis. These could be the plots at which we measure biodiversity. And then we have the extent, which is the area that covers all the plots, all the grains. Right? So we have grain and extent. The grain is in black here, and extent is in gray. Now we can change the grain of the study by, for example, increasing the size of the plots at which we sample biodiversity. We can increase the grain. Or we can decrease the extent, as over here. So instead of studying the plots worldwide, we will study them only in Europe. Now, you can use the same logic to define phylogenetic scale as well. Grain is the smallest unit of analysis. For example, we study niche conservatism within genera, families, or classes. These are the smallest unit of analysis to study niche conservatism. Or we can keep the grain unchanged, so we study niche conservatism in genera, but we change the extent of the study. So we study genera in hummingbirds, birds, or vertebrates. What will be interesting for us in this talk is not niche conservatism as the variable of interest, but the diversity gradients. Right? So in this case, the grain will be the species, and we will change the phylogenetic extent of the study. Right? So we study gradients within hummingbirds, birds, and vertebrates. The null assumption here, which is often implicitly invoked, is that diversity gradients stay unchanged across the phylogeny. So if I study hummingbirds, birds, or vertebrates, I will in principle get the same results. Now there might be some increase in gradient variation as I move from the root toward the tips of the phylogeny. So as I move across the phylogeny, I may get more variation in the gradients, but there should be no change in the average. On the average, the gradients stay the same. We saw that even though higher taxa show the famous gradients, there is a lot of variation. Here we have the gradients depicted for mammals and here for birds. And you see that most of the clades have the peaks of diversity in the tropics, but there's lots of variation. Some gradients can be flat, as over here, and the extreme example would be the exceptions to the rule, such as salamanders or geese, which have peaks of diversity outside the tropics. So maybe we can study this variation in the gradients to achieve a more general and a more comprehensive picture of the gradients. What the gradients look like. Can we capture that variation as well? And in doing that, we can also test the alternative to the null assumption, namely, are there any phylogenetic trends in the gradients? Do the gradients change in a systematic way as we move across the phylogeny? Are they phylogenetic scale invariant or phylogenetic scale dependent? This is a research topic that we investigated within the working group, which was organized by IDIV. There have been multiple meetings in Germany, in Prague, and in Switzerland. And one of the projects that came out of this group, which I'm leading, is investigating diversity gradients across phylogenetic scales. Here we see the results depicted for birds and for mammals. You see these circles which have different colors, and the color coding corresponds with the slope of the latitudinal gradient, right? So you plot diversity against latitude, look at the slope of that relationship, and the slope is then plotted for all the different clades. In blue, we have the classic gradients. Most species are in the tropics, few species in the temperate. In red, we have the inverse gradients, and then the yellow are the gradients which are flat somewhere in between. So you see that when we're at the base of the phylogeny, we see the classic gradients. But as we move toward the present, there is a lot of variation in the slopes of the gradients, including reverse gradients, flat gradients, and there is a significant increase in gradient variation. The same results are plotted over here. This is the slope of the gradient, and this is the age of the clade, or the age of the taxon. And you see that there is an increase in variation, but it's interesting that there is not only an increase in variation, there is also a change in the mean. So we see that there is a phylogenetic trend. And this phylogenetic trend shows that the old and large clades show strong gradients, whereas the small and young clades show weak gradients. So the gradients change as we move across the phylogeny. Also, these phylogenetic trends that I just described are consistent across birds and mammals. So they seem to be like more consistent across different taxa. I investigated this in more detail by correlating latitude with different, correlating diversity with different predictors. So how the diversity changes with the latitude, how it changes with temperature, how it changes with environmental productivity. The gradients can also be measured using the slope of the relationship or the slope of the relationship after the log transformation using correlation coefficients. And you see that even though these are different setups, some similarities emerge across all these panels, namely that there seems to be a phylogenetic trend where the old and large clades show strong gradients, small and young clades show weak gradients. Let's have a look in detail at one example. For example, the relationship between regional diversity and regional productivity. When you look at the slope of the relationship, you see that it changes with the number of species in the clade. So there is a pronounced phylogenetic trend. This pronounced trend, however, results necessarily from the clade diversity itself, right? So when you have a large clades and you go along the gradient, you are adding more species across the different parts of the gradient because the clade has many species as compared to a small clade. So to account for this issue, you can use correlation coefficients instead. So instead of studying the slope, we can study the correlation coefficients. And you see that the trend becomes weaker, but it's still there. Right? So the trend is still there. I have also done some simple null modeling, which you see the null model is depicted here in the light green, where you keep the phylogeny constant, keep the configuration of species ranges constant, but you reshuffle the names of species on the phylogeny. And you see that even this simple null model can reconstruct the trend, which actually captures much of the variation that we see in the data. So the trend itself, the phylogenetic trend, seems to be largely non-biological, right? It emerges even within a simple null model that only considers the shape of the phylogeny and the configuration of species ranges in geographic space. So what we have seen is that the trends depend on how the gradients are measured. If we use slope, correlation, temperature, latitude, and so forth. And the trends arise even under the null model, meaning that they are largely non-biological. I will not go too much into the technical details here, but I will briefly summarize that diversity gradients seem to be far from universal. In fact, 36% of clades within mammals and 37% of clades within birds show inverse gradients. Right? So we found that these inverse gradients are surprisingly common. When you actually look at the data, they're surprisingly common, and they also occur in relatively large clades. So to be absolutely sure that the clade does not show the inverse gradient, you have to pick a clade that has over 500 species, right? So as long as the clades are smaller than 500 within birds and mammals, you will find that the inverse gradients occasionally emerge. Consequently, we can reject the null hypothesis that diversity gradients are unchanged across the phylogeny. And this has a potentially relevant implication. The implication is that when you're choosing the taxon for your research, when we're choosing the taxa that we're going to study, the age and the size of the taxon and the measure of the gradient that we choose predetermine to a significant extent the shape of the gradient. Right? So if I choose to work with hummingbirds, birds, or vertebrates, the choice of the taxon and the implicit choice of the taxon age and taxon size predetermines the shape of the gradient that I'm going to get, and maybe to some extent also the results of my study. Now, I will find differences in the gradients of hummingbirds, birds, and vertebrates, but these differences do not show that there are different causes, biological causes, behind the gradients. It's simply a statistical necessity which follows from the shape of the phylogeny and species distributions. So this would be a proof of concept, which shows that phylogenetic scale is something that we might need to consider in biodiversity research, much like we consider spatial scale and temporal scale. So we have explored working systematically across scale, and finally, we can move to the research linking diversity dynamics to the mechanisms that generate the dynamics at the microevolutionary level. Why are the mechanisms important, right? Because knowledge of the mechanisms will provide us with deeper insights into the causality, right? Like the mechanisms are associated with causality, what causes the differences in biodiversity. And once we understand the causality and the processes that generate diversity, we can better predict diversity, for example. And the mechanisms that generate diversity, such as speciation and extinction, have been studied extensively at the level of microevolution, right? So we typically say that diversity, the mechanism generating diversity, is speciation. But speciation itself is a result of other mechanisms unfolding within the realm of population genetics. So maybe there might be some principles that extend from population genetics, the evolution of reproductive isolation barriers, all the way to speciation extinction and to the dynamics of diversity. So maybe the microevolutionary dynamics of speciation, the generation of diversity, can explain some of the diversity that we see at large scales. We explored this question in a working group at the University of British Columbia. Namely, we tried to identify those mechanisms that extend across scales from micro to macroevolution to answer the fundamental question of how is diversity generated. In a paper that we published, it's a perspective piece, we identify some empirical and conceptual similarities between micro and macroevolution that might be potentially promising. The empirical similarities involve the fact that evolutionary rates depend on the time interval. This is a relatively new and surprising finding, right? The evolutionary rates depend on the time interval. We can see this depicted in the figure here. So we have the evolutionary rate and the time in millions of years. And you saw that, you can see like this is the absolute scale and this is the log transformed scale, time and evolutionary rates. And you see that when we study evolution over long time intervals, especially in the deep past, the evolutionary rates seem to be slow. But when we study evolution over short time periods, typically toward the present, evolution seems to be fast. Now, it's interesting that this relationship, this negative relationship, emerges not only for the rates of molecular evolution, but also for the rates of morphological evolution, and at the macro scale, for the diversification rates inferred from the phylogeny and from the fossil record. So we have some similar relationships across micro and macro evolution. Maybe we have some scaling laws in evolution, which suggest altogether that evolution is fast in the short term, but slow in the long term. And these scaling laws seem to apply below and above the species level. On the conceptual side, a potentially useful idea might be the evolutionary landscapes. In microevolution, we have the concept of the fitness function and the adaptive landscape. The fitness function captures how fitness changes with the trait value of individuals, right? So this is an evolutionary landscape at the individual level. We can also average the trait values across individuals in the populations and get the mean fitness to get the adaptive landscape. So this adaptive landscape emerges from averaging the fitness functions across the individuals within populations. So we can stock these landscapes at top of each other at the level of individuals, populations, and also potentially of species, where we often see that traits evolve towards certain optimum values, right? The optimum distribution of beak sizes in Darwin's finches, as identified by the Ornstein-Uhlenbeck model on the phylogenies. So this would be the level of species, and finally, the level of entire clades, where diversification rates, which can be thought of as a fitness under the species selection concept, the diversification rates also change with traits. So here we have the level of individuals, populations, species, and entire clades. And you see that the effects of traits on fitness seem to translate from one level of organization to the next. The conclusion here would be that the empirical and the conceptual similarities, maybe some of these that I have just briefly outlined, might help connecting across scales the mechanisms that generate species diversity. This general topic is also something that a student that I co-supervise in Prague has recently published on in biological reviews. What Mohammad is interested in is the mechanisms of sexual selection in plants. And what he argues in this paper is that maybe we can identify some traits which capture the strength of sexual selection, and we can identify some traits which capture the strength of sexual selection, and we can use these to predict macroevolution. And in the paper, he proposes several such promising traits, such as the pollen-ovule ratio, right, or the style length. These traits, the pollen-ovule ratio or the style length, they capture the competition between different males for the fertilization. So there's pollen from different males ending up on the style and then growing into the style to fertilize the egg. So there's competition between males, the strength of sexual selection, and there is some indication that these mechanisms of sexual selection correlate with diversification rates at the macroevolutionary scale. So this is just one example of how to explain the macroevolutionary dynamics, the mechanisms that generate diversity at large scales across the phylogeny in the geographic space, might need to be explained using microevolutionary mechanisms that generate new species. Now I understand that these are perspective and conceptual papers, so maybe I will give you an empirical example, like to make it a little bit more alive. One empirical example that we have investigated was the co-speciation in figs and their pollinators across elevations. This is a study that we developed together with my colleagues from České Budějovice. And we studied the figs and the fig wasps that pollinate them. So this is a hallmark system for the study of coevolution and co-speciation, right? Because there are two interacting partners, the plant and the pollinator, and they depend on each other for reproduction. So there is a very tight coevolution, one-to-one matching between the species of plants and the wasps. At the same time, we know that there are quite many species of fig wasps and quite many species of figs. So it seems that there is significant diversity, which is often attributed to the co-speciation and coevolution. But because of this one-to-one matching, we don't really know how the speciation, the co-speciation unfolds. This is a question that my colleagues decided to address getting some data on population genetic structure for 6 species pairs along the elevational gradients in New Guinea. So here we have the figs and the wasps and the elevation gradient, and they had some hypotheses about how the populations would differentiate during the speciation and along the elevational gradient. And of course, we found that these hypotheses are more or less supported. One result which was surprising, but intuitive in the hindsight, was that the fig wasps differentiate and speciate faster than the host figs, right? So the differentiation within the wasps is faster than the differentiation within the figs. And these differences temporarily break down the one-to-one matching. So there is the breakdown of the one-to-one matching, one wasp, one fig. However, as the wasps are differentiated, this triggers the differentiation within the figs, which eventually speciate as well, which restores the one-to-one matching. So it is this cycle of the breakdowns and restoration of the one-to-one matching and this process of coevolution which generates the diversity of both, arguably, the figs and the wasps over time. This brings me to the conclusions. Tropical diversity has generated biologists ever since Humboldt and Wallace. But we still do not have the definitive answer to the question, why are the tropics megadiverse? This is limiting a synthesis which we need to advance the theory of biodiversity, and the synthesis would also be useful to reconcile the conflicts in previous work. Synthesis would also be useful to predict changes in diversity and to efficiently safeguard diversity. However, even though we don't have the synthesis yet, there might be some potentially promising research avenues toward the synthesis. And today, I tried to explore 5 possible ways to advance the synthesis and show you some examples of how these ideas could be practically implemented. We saw that we can study the different explanations for the tropical megadiversity. We can study them together. We can investigate the interplay of mechanisms behind the explanations. We have seen that maybe we can focus on studying diversity patterns in general, across elevations, latitudes, but also focusing on the exceptions to the rule. We can work systematically across scales and maybe link the dynamics of diversity to the microevolutionary mechanisms generating the diversity. By walking down these research avenues, we have learned that multiple mechanisms seem to operate simultaneously: time, diversification rates, and energy limits, and none explains the diversity gradients completely. We have also seen that sometimes the same gradients may have been generated by very different mechanisms. In the case of geese, albatrosses, cetaceans, birds, and mammals, as revealed by the exceptions to the rule. These conclusions might hold also for the regular gradients, not only for the inverse gradients, and they suggest that there is no one correct answer. However, some generalities seem to emerge. At least in the birds in the neotropics, it seems that the mechanisms generating diversity tends to interact with each other in a consistent manner. We saw that diversification tends to decline over time as the clade approaches the energy limit within the region. So the diversification declines, the accumulation of species declines over time as well, but despite these energy limits, new species continue to accumulate everywhere, especially in the temperate. Right? So we see the inverse relationship between diversity and diversification. And maybe some deeper insights into the causality might result from studying the microevolutionary dynamics which fuel the generation of new species. Those were the examples of the mechanisms of co-speciation, sexual selection, and the scaling laws. In future work, to identify the causes of tropical megadiversity, it therefore seems to be potentially useful or potentially promising to study the mechanisms together, to study the exceptions to the rule, and to consider the scale of investigation explicitly, especially the phylogenetic scale, which remains understudied. So we see that even though we do not have a definitive answer to the question, why are tropics megadiverse? In attempting to answer this question, it seems that we are beginning to uncover the general principles that govern the evolution of diversity. right? So we're beginning to understand these general principles that govern the evolution of diversity, thus building on the foundation of the biodiversity research laid down more than 100 years ago on the evolution of diversity that has fascinated biologists ever since Humboldt. Thank you very much.
Hi, thank you. This was really inspiring. Very quickly, regarding the first part of your talk, so you focus on birds, and I'm assuming, or I think I heard that there were birds that are occupying different trophic levels. But I was wondering whether you can extend on this. What do you think? What role do trophic levels play in this story? Like, would the patterns be very different across, or is it sort of similar?
I think one of the exciting things for me was that we find similar results despite the fact that these birds are at the different trophic levels. But maybe if we had this information and included it into the analysis, we would get even more consistent findings. So considering the trophic levels, the traits, considering more information about the species, that would definitely be the next step. Thank you for the suggestion.
Okay, thank you a lot. It's a great talk. I was wondering, at the beginning you talk about energy diversification rate and the time, but you didn't consider biotic interaction, because we can imagine that if you have many species with whom to interact, you have higher chance to speciate and find new niche. So is it possible to integrate this at the beginning?
It would be amazing. Like, I was thinking about it long and hard, but I couldn't figure out a way to do it like practically. And definitely it would be amazing to include the biotic interactions in some compelling way. I'll spend some more thoughts. Like, if you have some suggestions or like know of some of the data that could be suitable, we can talk over coffee. That would be really cool. Like, one thing, just one insight about the diversity begets diversity. It's an explanation which works, but to some extent you already have to assume that there is lots of species in the tropics to explain that there is more species in the tropics. So that's an interesting way to think about it as well.
Okay, thank you. Very interesting presentation. I'm just wondering how temporary events such as climate change and anthropogenic effect affect the pattern of diversity. Thank you.
That's a really good question, right? Because all of these groups have experienced the glaciations, the changes in the global climate, which have been relatively recent, in the last couple of 100,000 of years. And you would expect that these processes, these changes have, for example, they have hampered the signal in the data that we can uncover. And it seems that the signal in the data here, right, like what has been happening over millions of years, was strong enough to be detected even despite all these events like the glaciations and changes in the climate. I feel that it would be a useful extension of the work as well, right? Thank you. I was trying to just stay as parsimonious as possible in these studies, but I completely agree, including biotic interactions, trophic levels, climate change, that would be amazing.
Yeah, thanks for a very nice talk. I was wondering, when you were talking about the energy, do you really need only energy? I mean, that's something that has been bugging me in all of these discussions about the diversity gradients, that it seems that most of the models are assuming kind of sunlight as the source of energy, and that's more or less what is assumed also as being at the base of the productivity gradient. But then actually the primary productivity depends on water availability to a very, very high degree, and also to nutrient availability. So these are, as far as I have seen, they are very rarely considered in these things, even though if we look at the map and look at the gradients, the dry areas tend to be the drier, the less species you have. And also, if you go to the very poor kinds of soils, very poor productivity, with some exceptions, you also start to get fewer species. So how do you think this should be factored in?
Maybe I don't know enough about soil and I need to study up, because I didn't see any clear patterns. There are different soils which we can there are some patterns in the data that we can't explain without considering the soil, but it simply might be my ignorance about the different soil types. Regarding the different measures of energy, I try to include environmental productivity, actual evapotranspiration, which also includes a little bit of the water availability. And it seems that these differences do not influence the results in a qualitative way, right? So maybe at these large scales, globally and over millions of years, these differences are not as relevant as, for example, at the local scales when studying the productivity-diversity relationship within the communities. But it's an important distinction.
Yeah, what it does do is that it causes some of those gradients that we're looking at. They are not really latitude, they're not really altitude, because there is this productivity which is not linearly related to either one of those.
Definitely, I agree with that, and I'll be thinking about it some more.
Yeah, I agree in most of the points that you raised, but I'm thinking a bit back and forth about the dualism of tropical groups and temperate groups.
Mm-hmm.
Isn't it that you have a particular challenge in the tropical groups and a particular challenge in the temperate groups?
Mm-hmm.
And if you look at, for example, the salamanders, I think you see the pattern of the challenge of the temperate group very well, because they are normally not capable to live under tropical conditions. And then you have one group, which is the Bolitoglossa, which entered from the center of diversity of salamanders, so the east of the United States, into the mountain areas of Central America. Here having a wonderful radiation into many, many species in Bolitoglossa, entering South America, and only one group, Bolitoglossa alt-amazonica, entering the tropical lowlands. And they are still so badly adapted to tropical conditions that there is a discussion whether it's one species or four Bolitoglossa alt-amazonica, but they are not well adapted to these tropical conditions and cannot compete. But if they really catch up and get competible, then there might be another radiation in Bolitoglossa in the tropical Amazon area. So maybe many things can be even better explained if you say there is two very different realms with very different challenges, the tropical one and the temperate one.
I would agree with that, definitely, right? So like maybe once these species unlock the key adaptations for the tropical life, they will start to diversify there as well. Thank you very much.
So we have nice group picture. Thank you.
Thanks for a very nice talk. So overwhelmingly data rich. Yeah. Great talk. I was about to ask, but the time was short.